DNA and RNA. So starting with what DNA stands for, deoxyribose nucleic acid. RNA stands for ribose nucleic acid, and both of these are super useful because it informs you of the type of sugar they each contain. So DNA contains deoxyribose, RNA contains ribose. Now the building blocks of DNA and RNA are known as nucleotides or nucleic acids. So what does a nucleic acid consist of? It consists of a sugar, as I've already mentioned. In DNA that's deoxyribose, in RNA that's ribose. It consists of a phosphate group, which is inorganic, meaning it doesn't contain carbon, and it contains a nitrogenous base, meaning it contains nitrogen. Now the bases vary depending on if you're discussing DNA or RNA. Nucleic acids or nucleotides join together through complementary base pairing. So what are the four nitrogenous bases in DNA? They are A, T, C, and G. A stands for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. In RNA, the thymine is replaced with uracil, U. If we look at the structure of the bases a little more closely, we can divide our bases into being either purines or pyrimidines. Now, the major difference between the purine and the pyrimidine is the size of them. Purines contain 2 rings of carbon atoms, whereas pyrimidines contain only 1. Examples of purine bases are adenine and guanine. Examples of pyrimidine bases are thymine, cytosine, and uracil. The size difference between the purines and pyrimidines means that hydrogen bonds can occur between the bases, and it's these hydrogen bonds which hold together, in effect, the rungs of the DNA ladder. Remember that DNA is double-stranded. It's a long molecule when compared with RNA, and in terms of the number of hydrogen bonds holding together the bases of DNA, it's 2 hydrogen bonds between A and T, 3 between C and G. Now those phosphodiester bonds occur between the deoxyribose sugar and the phosphate. Now the type of reaction which occurs for a phosphodiester bond to form is known as a condensation reaction, which we've met many times before. This means that it involves the loss of a water molecule. It makes sense, therefore, that the type of reaction which breaks a phosphodiester bond is known as a hydrolysis reaction, which involves the use of water. So what are the roles of DNA and RNA? So DNA stores all the organism's genetic information, and it codes for the sequence of amino acids known as the genetic code. RNA's role is slightly different. It transfers genetic material from DNA to ribosomes. Now, we know that ribosomes are where protein synthesis occurs, but what are ribosomes made out of? Well, they're made out of a combination of protein and ribosomal RNA, rRNA. They're made in the nucleolus of the nucleus. If we look more closely at how the structure of DNA makes it suitable for its role, first of all, it's a polymer, a very large molecule which contains a lot of information. There are hydrogen bonds between bases. Now, those hydrogen bonds are relatively easy to break, and that makes it easy to separate the strands of DNA. The DNA is double-stranded, as you see here, 2 strands, and that means that each strand can act as a template for replication, which we'll look at very shortly. The strands are anti-parallel, which means they run in opposite directions, and this allows the double helix to twist and provide a compact shape. That's important. We need it to take up very little room in our cells. And then finally, the complementary base pairing I mentioned allows DNA to be replicated without error, reducing the frequency of mutations. Remember, a mutation is a spontaneous and rare change in DNA. So looking more closely at the structure of DNA, as I've said, double-stranded, the DNA folds itself up into a double helix. You've got your sugar phosphate backbone. Here's your deoxyribose sugar. It's known as a pentose sugar. Look at the shape of it. That explains why. So here's your sugar phosphate backbone. Phosphodiester bonds hold together the sugar and the phosphate groups. If you have a phosphate and a sugar and a base, we have what's known as a nucleic acid or DNA nucleotide. And then we have hydrogen bonds that hold together the complementary base pairs, 2 hydrogen bonds between A and T, 3 hydrogen bonds between C and G. And remember I said the pyrimidines include thymine, cytosine, and uracil. They consist of 1 ring. Purines, which include adenine and guanine, consist of 2 rings. Let's focus in on this anti-parallel strand a little more. So that means that the 2 sugar phosphate backbones of DNA run in opposite directions. We use the 5' and the 3' notation in order to represent that. And all that really refers to is the carbon on the deoxyribose. So here's our deoxyribose sugar, and we know that the fifth carbon exists up here. So that's in effect up there. And then because of the way that strand runs, it means that at the bottom here, we have the third carbon, as seen here. Because this strand is running in the opposite direction, that therefore means that that top carbon is the fifth carbon, and this bottom carbon here is the third carbon. Today we're going to look at semi-conservative replication. Now this is the mechanism by which DNA is copied, and actually what happens is that each new DNA molecule formed contains one old strand and one new strand. And we're going to actually look at how that replication occurs, as well as the various experiments that proved this mechanism. So semi-conservative DNA replication states that DNA helicase, which is an enzyme, breaks the hydrogen bonds holding together the complementary base pairs in DNA. Now, each of the DNA strands acts as a template. There are 3 nucleotides present in the nucleus, which can then pair up with the exposed bases on DNA. In order to produce the new strands, obviously we need to pair up the exposed bases, and again we need hydrogen bonds in order to do that. Remember, our complementary base pairing rules apply, so where you have adenine, thymine will bind, where you have cytosine, guanine will bind. Adjacent nucleotides are joined together through phosphodiester bonds, and it's the second enzyme, DNA polymerase, which is responsible for forming these bonds. Now, 2 new strands of DNA are formed which are identical to the original DNA, but as I've already said, semi-conservative replication means that each DNA molecule contains one new strand and one old strand. So how was the theory of semi-conservative replication proved? Well, it was through the experiments carried out by Stahl. They grew E. coli bacteria in what we call heavy nitrogen. This is nitrogen with a mass number of 15. Then the sample of DNA was isolated using a centrifuge, which is in effect a machine that spins the DNA very quickly. That bacteria was then grown in a second nitrogen medium, but this time containing a lower mass isotope, 14 nitrogen. Now that DNA was isolated by a centrifuge after 1 replication cycle, after a second replication cycle, and after a third replication cycle, and the density of each DNA sample was measured. What was interesting was that after the first replication sample, the DNA was of an intermediate density, and that therefore meant that after 1 replication cycle, that DNA contained 1 heavy 15 nitrogen strand and 1 lighter 14 nitrogen strand, i.e. the DNA contained 1 old strand and 1 new strand, because it made the new strand from the lighter nitrogen. So after 1 replication cycle, all the DNA was of an intermediate density. After the second replication cycle, 2 bands of DNA were seen, one of intermediate density and one of light density. And then after the third replication cycle, the lighter density band contained more DNA. This is our original parent strand of DNA. As we know, it was grown in only heavy nitrogen. So both strands contain heavy nitrogen, and our centrifuge results shows that heavy density because all the DNA is that heavy isotope. Whereas after the first replication cycle, when it was grown on the lighter nitrogen medium, because of semi-conservative replication stating that each new DNA molecule will contain 1 old strand and 1 new strand, you'll expect to see bands of an intermediate density containing 15 nitrogen and 14 nitrogen. As the second replication cycle takes place, again grown on that lighter nitrogen medium, obviously semi-conservative replication states that you'll expect 1 old strand and 1 new strand in that DNA, but because previously it was grown on lighter nitrogen, this is why you have these 2 DNA molecules containing the lighter nitrogen only, and that's represented by this lower density band here as seen on the centrifuge results, as well as the 15 nitrogen and the 14 nitrogen, which occurs due to these 2 DNA molecules. And then so on and so forth as you go through the third replication, more light nitrogen. This will increase the amount of that lighter density DNA, so that band here gets thicker and the heavier band takes up less of that centrifuge result. And then from my guide, I'm really hammering home this point that the Stahl experiment proved that DNA was replicated in a semi-conservative manner. And do just make sure that you read through these notes. The other 2 theories of DNA replication are also mentioned here, which is the conservative replication, which predicts that both heavy DNA and light DNA will be present, but no intermediate density will be present, as well as the dispersive replication model, which predicts that after one replication cycle, the density of all DNA molecules will gradually become lower, so no intermediate density DNA should remain after the second replication cycle. Because remember, the dispersive replication model basically says that all the DNA nitrogenous bases will be randomly jumbled up. The diagram shows part of a DNA molecule. How many nucleotides are shown in the diagram above? I'll mark them off. Remember that a nucleotide is a phosphate, sugar plus base. 1, 2, 3, 4, 5, 6, 7, 8. The answer is 8. Name the type of bond labelled X in the diagram. So it occurs between our sugar and our phosphate, so it's a phosphodiester bond. Describe the structure of DNA. Well, it's a polymer of nucleotides, which consist of, you've guessed it, of phosphate, sugar, base. Complementary base pairing occurs between bases. So you've got adenine joining to thymine, cytosine joining to guanine. Bases join through hydrogen bonds. Don't forget to point out that it's a double helix. And to add an extra detail, we would point out that the nucleotides join in phosphodiester bonds. The diagram below shows part of a DNA molecule. Name the type of bond between complementary base pairs. That's hydrogen bonds. Adjacent nucleotides, that's going to be phosphodiester bonds. The length of a gene is described as the number of nucleotide base pairs it contains. Use the information in the above diagram to calculate the length of a gene containing 4.38 times 10 to the 3 base pairs. So just count this, 1, 2, 3, 4, 5. We've got 5 base pairs here. So we need to do 4.38 times 10 to the 3 divided by 5 and then multiply it by that length of 1.7 nanometers. So that's 1,489. Complete the table below with ticks to show which elements are found in the following biological molecules. So we have galactose, which is a carbohydrate. So remember I said CHO, carbon, hydrogen, oxygen. Phospholipids, lipids, yes, again, carbon, hydrogen, oxygen. However, you've also got phosphate present in that phosphate head. So I'm going to add a P. RNA is a nucleic acid. So we need to say carbon, hydrogen, oxygen. Remember, RNAs have a nitrogenous base, and there'll also be phosphate in that sugar phosphate backbone. Sucrose is a carbohydrate, so it's just CHO again. So just tick these off nice and carefully. After Watson and Crick proposed the model of DNA structure, scientists investigated the possible mechanisms for DNA replication. 2 scientists grew a bacterial population, providing them with a nitrogen source containing only heavy nitrogen, 15N. As soon as all the DNA in this population contained 15N, the scientists changed the nitrogen source to one containing the lighter isotope, 14N. They changed the source at 0 hours. During the investigation, the scientists measured the size of the population of bacterial cells. Figure 1 shows the scientists' results. The generation time for a population of bacteria is the time taken for all the bacteria to divide once by binary fission. Use figure 1 and the following equation to calculate the generation time for this population of bacteria. Give your answer in hours. Now, considering this is only 2 marks, this is an awful lot of work and a pretty difficult question. The first thing to look at is what's here. It says size of population at time plus 4 hours, size of population at time minus 4 hours, which means I need to just draw a couple of lines here and here to work out the population sizes at the relevant times. So it's here and here. The most difficult thing now is obviously reading this scale. So just remember, 10 to the 5 just means 1 times 10 to the 5. This means 1 times 10 to the 6, and so on and so forth. So as you read up the scale, this light grey line here is 2 times 10 to the 6, 3 times 10 to the 6, 4 times 10 to the 6, 5 times 10 to the 6, 7, 8, 9, 9 times 10 to the 6, and then that's how we reach 1 times 10 to the 7. So that is 1 times 10 to the 6. Let's do the same up here. 1 times 10 to the 9, 2 times 10 to the 9, 3 times 10 to the 9. So I can now start subbing in my values. Number of generations is log to the base 10, 3 times 10 to the 9, divided by 4 times 10 to the 6, divided by log to the base 10 to the 2. So you just need to use your calculator really carefully here. And what that will tell you is that you have 9.55 generations, but just keep an eye on that time frame. This, remember, was a total of 8 hours. So how do we work out the generation time? We just need to do 8 divided by 9.55 to get 0.84 hours per generation. Really stinky question. At intervals during this investigation, the scientists removed samples of the bacterial population, isolated the DNA, and measured the density of the DNA. DNA made using 15 nitrogen has a higher density than that made using 14 nitrogen. There are 3 possible models of DNA replication. These models are shown in figure 3. Remember that the 3 models are conservative, semi-conservative, and dispersive. Now, figure 2 shows results possible from a semi-conservative replication, because remember, semi-conservative replication involves the new DNA molecule containing 1 old strand and 1 new strand. And so you can see that after 1 replication cycle, you can see that generation 1 contains DNA which is all of an intermediate density. Then after the 2nd replication cycle, generation 2, we can see 1 intermediate density and 1 light density. And then as we move to generation 3, we can see the lighter density band contains more DNA. So that all fits with a semi-conservative replication style. And therefore, look at Q. These new strands in generation 1 contain 1 old strand and 1 new strand. Yes, that fits semi-conservative replication. And as the generations move on, we get more lighter nitrogen present. So it's model Q supported. We know that this is semi-conservative replication. So why don't the results support P? Because in generation 1, you can see that you'd expect a heavy density and a light density peak, which is obviously absent. And why is this model not supported by the results? Because in this dispersive model, you'd expect many more peaks in the 2nd and 3rd generation, which we don't have. Draw and label a single DNA nucleotide, so we know we need a phosphate. We need a pentose sugar. In DNA, remember, that's deoxyribose. And we need that joining to a nitrogenous base, which is in DNA either A, T, C, or G. Give 2 features of DNA and explain how each one is important in the semi-conservative replication of DNA. Right, in order for semi-conservative replication to occur, we need the new DNA molecule to contain one old strand and one new strand. We need nucleotides to pair up with exposed bases. So first of all, we need to break the hydrogen bonds in the original DNA double helix. So features include DNA must contain hydrogen bonds between bases which are easy to break. And secondly, we need to state that DNA must contain two strands, both of which can act as a template. Replication of mitochondrial DNA is different from that of nuclear DNA. The replication of the second strand of mitochondrial DNA only starts after 2/3 of the first strand of mitochondrial DNA has been copied. A piece of mitochondrial DNA is 16,500 base pairs long and is replicated at a rate of 50 nucleotides per second. Tick the box that shows how long it would take to copy this mitochondrial DNA. Okay, now this is another difficult question. So these lines here represent your double strand, and it says that the replication of the second strand of mitochondrial DNA only starts after 2/3 of the first strand of mitochondrial DNA has been copied. So that's what I'm showing here. If this is in essence one single strand, the second strand only kicks in after the first 2/3 of the first strand have been copied. And then obviously that first strand is completed, and then there's the final 2/3 of the second strand to complete. So in effect, we have 16,500 base pairs multiplied by, add these up, 5/3, and then in order to work out that time, you need to divide it by 50 because it tells us that it replicates at a rate of 50 nucleotides per second, giving us a grand total of 550 seconds. Okay, question 4. Describe how a phosphodiester bond is formed between two nucleotides within a DNA molecule. Okay, this is a more straightforward question. It is important that you bank these 2 marks because I'll show you the next part, which is gross. So it's going to be a condensation reaction, which I've specified involves the loss of a water molecule, and this enables the phosphodiester bond to form between the phosphate and the deoxyribose. The 2 DNA strands of a particular gene contain 168 guanine bases between them. The relationship between the numbers of guanine bases G, adenine bases A, thymine bases T, and cytosine bases C in these 2 strands of DNA is shown in the following equation. Use this information and your understanding of DNA structure to calculate the maximum number of amino acids coded by this gene. Horrible question. Remember, you need 3 bases for each amino acid. That's important. Because of the way complementary base pairing works, we know therefore that we have 168 cytosine bases which pair with these 168 guanine bases. As I've already said, because adenine binds to thymine, it means that you have the same number of adenine bases as thymine bases, which means we can sort out this equation a little bit to give us fewer unknowns. I'm going to sub in T for another A based on that, and then just sort this out a little bit. We know some values, so we know 168 is our number of bases of guanine and cytosine. So if we solve for A, we know that we have 42, which means by definition the number of thymine bases is also 42. Only one strand of the DNA is transcribed, so we actually only want to look at one DNA strand when we look at the number of potential amino acids. So in effect, we need to halve all the bases we've just calculated. So that means we have 84 cytosine bases, 84 guanine bases, 21 adenine bases, 21 thymine bases. So add those 4 numbers up in total, which means we have 210 bases, and then finally divide it by 3 to find the number of amino acids. So that's 70, an awful lot of work again for just 2 marks.